So we have three various areas that we work in the lab. One is the microbiome and effect of microbiome in behavior and the behavior that manifests by autistic trait. The other area of our work is to test the various drugs to see how we can improve the synaptic transmission. And this is the part we're going to talk about today. And we also have some work about the auditory processing disorders in animal and also in human clinical trial. So let's talk about mGluR5 receptor. I don't know if you have heard about this receptor, but it's a receptor that's very involved in various aspects of autism. So as you know, there is various types of receptor. They are ionotropic or metabotropic, and mGluR5 is a metabotropic receptor. And generally ligand-gated ion channel directly allow the transfer of ions, but in case of metabotropic, that require a G protein and a second messenger to change the transmission. So this is our mGluR5, and glutamate is bonded to it. Is a gray in the middle of the glutamate, as you can see it. So that's the receptor we're going to talk about today. So mGluR5 belongs to the group 1 metabotropic receptor and is located in neurons and astrocytes. Is linked to NMDA receptors and to GABA. And you know that GABA is a main inhibitory neurotransmitter in the CNS. Yesterday, Dr. Frey mentioned about those different neurotransmitters, and I do not want to go to the detail because of the limited time we have. So it's implicated in plasticity and neural inhibition through GABA, and utilizes glutamate to strengthen neural connection, and is involved in long-term potentialization. Here is our glutamate, as you know, is mainly excitatory transmitter. The highest concentration is in CNS, and specifically in the forebrain, striatum and limbic regions. And it is essential, mGluR5 is essential in regulation of the glutamate. And is involved, as you know, in autism, Fragile X, tuberous sclerosis. So mGluR5 is coupled to the GQ signaling pathway. And there is two pathways mainly involved. One is the protein kinase that involve MAP kinase and mTOR. As you know, the protein kinase pathway lead to the alteration of the transcription of various proteins. And MAP kinase regulates transcription, proliferation, differentiation, and apoptosis. There's also activation of phospholipase C that involve calcium channels. So I think the video that I'm going to show you, there is a red dot, that's a glutamate, that bond to the receptor. The receptor dimerases. GTP is phosphorylated to GTP, and the gamma subunit dissociates from the alpha subunit. The gamma subunit activates phospholipase C and cleaves IP3 from DAG. IP3 travels to the endoplasmic reticulum, and calcium is released. This can cause either release of neurotransmitter or inhibition of the release. And calcium at the higher concentration of the DAG cause phosphokinase C to activate the MAP kinase signaling, which influence transcription. So let's look at the video. You can see that glutamate bind, dimerization, GTP become GTP, phospholipase C is activated, IP3 come to the endoplasmic reticulum, calcium is released, phosphokinase C is activated, and we have effect in the transcription, as you can see. So this is a diagram of the glutamate receptor, highlighting the main active site, as you can see, the glutamate in blue, where glutamate binds. And a separate allosteric site is there, and the ligand can activate that receptor. And that's what we're going to try to perform during our experiment, to activate this receptor through that ligand. So this diagram shows the protein that are involved in autism. You can see some in orange or red. The diagram shows that the proteins that are involved, the NMDA, mGluR5, AMPA, Chang3, and phosphokinase. This also shows the same as the last slide, but it's clear. It also shows the TO pathway that is involved. So the striatum we have seen yesterday, control movement and the human reward system, and mGluR5 protein are highly expressed postsynaptically in the cortex, striatum, and hippocampus. And in the physiopathology of ASD, one hypothesis is that mGluR5 activity is significantly decreased. So now we're going to show you a little bit of some of the work we have done, and then the work that is in progress. We published that recently. mGluR5 knockout rats exhibit a deficit in sociability. As you can see, in the rat that doesn't have this type of behavior, the white rat, they spend more time when they are using the three-chamber experiment with another rat that is already there. But when you have a knockout rat, the time spent here, as you can see, is similar between the stranger and the empty cage. So let's look at that. So we have here a rat here. We put our wild type rat. This is empty, and look what happened. The rat come in, spend time with the rat that is already in the cage. Rats are very social, as you know, and that's a very good model for us to see what happened with this aspect. And now, look to see catnap. That's another model we have in the lab. This is a catnap tree, and in this model, you see the rat comes, but it doesn't go there. This rat has autism and doesn't care about the other rat. Look around, change, go away, doesn't spend time in the cage that has another rat. So this is one of the ways that we measure the sociability of this rat, the time spent in each side. So now we can quantify all of this time spent in different area, and that helps us to understand what's happening. There's another area with the autistic rat is the social novelty, and as you know, we are all interested with the new person around when you go to a meeting. Actually, when I was preparing this talk, I was surprised. I thought maybe I'm autistic myself because sometimes I'm too shy to go to talk with new people. But it seems that the normal attitude is to go to new people. And rat shows that when you put another rat in the third space, the wild rat, that are the neurotypical rat, gonna go and check the new one. But the autistic rat doesn't do that. So let's see if the video is playing. So you see that this rat is a white type, go to the new one. Oh, what's going on? There's a new guy here, and spend time with both, okay? And now let's see what happened. When we do the same experiment with our autistic rats, we see that the autistic rat doesn't spend time with the new one. So I'm gonna go faster so we save some time for the other data that we have. This data shows that actually the wild type rat treated with the antagonist of the mGluR5, MTEP, exhibit ASD-like phenotype, and this exhibition of this phenotype is actually dose-dependent. So more you increase this antagonist, the more you're going to see that the rat present those type of behavior. And also some post-mortem human autism brain studies have shown that mGluR5 was found to be decreased in individuals with autism. So the objective of the specific presentation of today is to see the efficacy of the mGluR5 positive allosteric modulator, PAM, to ameliorate the autism-associated behavior deficit in autism transgenic rat, MECP2 knockout rat. Those rats have manifested some autistic trait. So just to remind all of us, what is the juvenile play behavior? The juvenile play behavior is a phenomenon that is displayed among juvenile both sex across many mammalian species. I have a new puppy at home. He's crazy with his juvenile play behavior. I cannot have any peace at home. So this is basically what happens to you when you have a new puppy or any new animal. So this is a very typical behavior. And as you can see here, those rats, the puppies should have those type of behavior. So let's look at their behavior. You see how? These rat are playing around, whistling and all of those things, okay? So this is another area that we characterize our animal models to see if those rats present those type of behavior. So when we quantify those type of behavior, we can see that social play behavior, diet in juvenile rat, is actually affected in knockout rats. So those knockout rats that we have in our study do not present those typical juvenile play, but once we give our CDPPB, we improve those juvenile play, as you can see here in orange. While if not, they are spending less time is in blue. So another type of characteristics that we look at our animal model is marble-bearing test. It's a simple, powerful behavior test. They represent anxiety and repetitive behavior, and it's easy to do, and it's easy also to score and to quantify. So that's another one, as you can see in this video. This is a wild type that doesn't have autism, and you can see that this rat does not seem to be anxious based on this test. Now, I don't have any other way to know, but the rat is interested in the marble, but does not borrow any of them. But then when you have an MECP2 rat, those, as you can see here, they're engaging in marble borrowing in the bottom right corner, and which is indicative of anxiety and repetitive behavior. Okay, that's just a short snippet of this video to show you how we do this test. And this is the same rat, but treated this time with our positive allosteric modulator, our medication, and you can see the rat stop doing those type of activity. So we basically try to characterize our animal model in the multiple different way of behavior that is characteristics of the core symptoms of autism, and trying to see if our treatment is active in most of those behavior. So here you can see that the quantification shows that the CDPPB treatment significantly reduced marble borrowing. So another aspect is sociability, and is impairment in social behavior is one of the hallmark of the ASD, and we can use again a 3-chamber apparatus, and it assesses cognition in the form of general sociability. So here, look at that. We have seen that a little bit before. This time we are using our MECP2 animal, the one that we're going to treat, and you can see that this animal came. This is a wild type. Check the other guy. Spend time with him, okay? And now let's see what happened to our autistic rat, MECP2. You see he come, look, but not very interested. Check around, and go the other side. There is nobody there. Spend time there, okay? And now let's see what happened with our new drug. So once treated with the CDPPB, you can see the rat going to spend more time with the other rat and stay in the same chamber. Okay? And when we try to look at you see the rat is spending time with the other rat. Happy there? Okay. So when we quantify that, we can see that the knockout rat that had the autistic characteristics, when treated with CDPPB, we can see that the rat spent more time in sociability. Now social novelty, as I mentioned before, we have a second rat in another chamber. I'm going to see if the rat goes with the social novelty. And you can see here that the white type spent time with the new one. You know, we add another rat here, and we see that the rat goes there to check the newcomer, spend time with the new rat, okay? And When we have a MECP2 rat, the rat is not interested with the other guy, looking at around, change, goes around, does not spend time. So we quantify the time spent in each chamber so we can analyze statistically if it is significant or not. And now we treat the same rat with the CDPPB, and you can see that the rat going to spend more time with the newcomer and become more social and spend time checking the newcomer and spending time to interact with the newcomer. So that's actually very interesting to see that how this rat can be a good model to test our new drugs. So we quantify all of those things, and then you can see that once we add the CDPPB, we have again, with all those animal treated, a normal pattern of behavior that we see in the wild type. Pre-pulse inhibition is another test that is actually very interesting because abnormal sensory processing is a hallmark of autism. I actually always thought it was not good that they didn't characterize the sensory processing as one of the core problems with the autism. And I see all the time with my patient that they have sensory disorders, sensory processing, and you know that with the patient with autism, this is one of the hallmarks. And I'm going to show you this pre-pulse testing inhibition that is very interesting. When in abnormal sensory processing, when a pre-pulse inhibition is when a startle reflex created by a loud pulse is attenuated by preceding by a weaker stimulus. You give a weaker stimulus, and then you give another stimulus, and you can see that the other stimulus, after a few times that you repeat that, neurotypical individuals are able to suppress or decrease the reaction to it. But autistic individuals are always overstimulated. They are not able to stop have an inhibition of this second sound. So let's see what happened with our animal. When we treat our animal with the CDPPB, we can see that even we are able to improve the sensory processing problem with these animals. And the knockout rat with no CDPP have an elevated startle reflex compared to white drop. Which means they are unable to adapt easily to the repetitive noise stimulus. But when we treat them with the CDPPB, we have a reduction of startle reflex and is very close to the wild type. So self-grooming. I think you are familiar with the self-grooming too. Self-grooming is a complex, repetitive, self-directed, and sequentially patterned behavior. A useful, I want to say, measure of repetitive behavior and anxiety. It's a very good tool that used in psychiatry usually to look at the treatment outcome. So it involves actually this diagram of a rat central nervous system and describe various areas. I think yesterday Dr. Frey shared with us very nicely how all of these different areas are involved in management of our normal behavior. But just to remind you guys that these various parts of the circuit are involved in just a small behavior like a self-grooming. Like it starts in neocortex, which is the area of the central nervous system, which will decide to initiate the behavior. Then we can see that the cerebellum modulates the coordination required for self-grooming behavior, the brainstem modulation, the pattern of grooming that will be seen. The hypothalamus regulates the neuroendocrine aspect of the self-grooming behavior. The amygdala regulates the context, specificity of the behavior, meaning of emotionally heightened state. It may play a role in initiating or inhibiting the behavior, and finally the stratum, which is important in maintaining the sequence of the grooming behavior. So a lot of circuits are involved. And let's see what happened with our animal that was treated with the CDPPB. We see that there is a reduction of excessive self-grooming significantly. So I think that's another interesting aspect that is managed by this treatment. So I would like to summarize our finding. This present study demonstrated that treatment of MECP2 knockout rat with the CDPPB helps in attenuating the ASD-like phenotype compared to vehicle or untreated animals. That was very exciting to see that we can have this beneficial aspect in various core issue of the autistic rat model that we have in our lab. Our results are in agreement with previous studies that demonstrate that the efficacy of mGluR5 agonist in ameliorating behavior deficit in mouse model. I think when you work in different animals, it's always good to see that you can reproduce the same effect in the various species before going to the human. These are the really translational research before going to humans, so it's good to know that we have a same reproduction of the same efficacy before we go to any phase 1 clinical trial. Our lab is actually specialized in various types of drug screening, drug development, from preclinical to the clinical stage. And we know that FDA requires significant amount of experiment in preclinical data to be able to, before to start the phase 1. So it's important to have 2 species, shows there's no toxicity and efficacy before we go to the phase 1. So we do not see any adverse effects of the CDPPB in animals. We didn't see any weight loss. We didn't see any other issues. No abnormality in the appearance of the animals. And this proof of concept studies is to explore the efficacy of the mGluR5 agonist in developing novel treatment modality for ASD seems to be exciting. So the future direction is to understand better the molecular mechanism involved in amelioration of the behavior deficit following the mGluR5 positive allosteric modulation. And we are also working on performing some neuroimaging to have more information about the neural correlates. So here is my team. I would like to recognize the work of Jamie Cooper. She did a great job. And Dario Ebodi, who was a postdoc from France. They did most of this experiment, and they helped to have this good result. And I would like also to recognize our sponsor. I'm happy to meet with you afterward and discuss more about this exciting result. Thank you.